Alright, in this video I wanted to discuss the difference between these terms haploid and diploid. Haploid and diploid are really important terms when we talk about the differences and similarities between mitosis and meiosis. We'll also be using these terms when we talk about genetics later on. So understanding haploid and diploid and how we end up with cells that have these characteristics is really important. So let's take a few minutes to think these terms over. So haploid and diploid, they both end in this word ploid, in this the ending ploid, and that relates to this word ploidy. So in genetics and biology, we use this word ploidy to describe the number of each kind of chromosome inside the cell. So what does that mean, right? We have these two words we're going to use commonly, haploid and diploid. Well, haploid describes a ploidy where the cell contains one of each kind of chromosome. Whereas diploid, diploid, I think two, diploid are cells that contain two of each kind of chromosome. So we can see that in this image. Haploid cells, here are three different kinds of chromosomes. We'll talk about telling chromosome kinds apart more later on, but basically we're telling them apart by size and shape and another characteristic. But there's one of each. So haploid cells contain one of each kind. Whereas the diploid cells right below, we have a pair of every kind of chromosome. So haploid, one of each kind. Diploid, two of each kind. When we talk about haploid and diploid, we might use this terminology N. So haploid cells are just N or 1N, whereas diploid cells are 2N. In this terminology N, and sometimes it'll be a lowercase n, but they mean the same thing, N is equal to the total number of kinds of chromosomes in the cell. So for this example on the right, we had 3 kinds. So in these cells, N equals 3. Again, we use the terminology N, 1N for haploid, or 2N for diploid, because we have two of each kind. So for these cells, the haploid cells contain a total of 3 chromosomes, because we're 1N and N equals 3. So our total number of chromosomes is 3. For the diploid cell, it has 6 chromosomes in it. And we can figure that out from knowing that diploid is 2N. And in this case, N equals 3. So 2 times 3 is 6. The cell will contain 6 chromosomes. All right, so that defines the terms haploid and diploid and how we use this N number to figure out how many chromosomes are in a cell. How can we determine how many kinds of chromosomes there are? So it turns out that biologists look at the chromosomes under the microscope, and they can tell the chromosomes apart. They can sort them into different kinds based on the chromosome's size, shape, and banding pattern. So here we can see a couple chromosomes in this picture. They have different sizes and shapes, and they also have different banding patterns. There's dark areas of the chromosome, and there's light areas of the chromosome. So biologists can take a cell, collect all the chromosomes out of the cell, stain them, and then observe them under the microscope. And based on their size, shape, and banding characteristics, we can sort them into different kinds. So we do that for human cells all the time. And when we do that, we generate what we call a karyotype. So karyotypes are just images that show all the kinds of chromosomes. So here we're looking at a karyotype, and it's a photo of all the chromosomes sorted by their size, shape, and banding. So in the upper left here, we have 2 copies of a chromosome that are the same size and shape, and they have the same banding pattern. So this is chromosome number 1. When scientists make a karyotype, they number the chromosomes in terms of by their size. So the largest chromosome will be chromosome 1. The next largest chromosome, this one, will be chromosome 2. Again, there are 2 of them. They have the same banding pattern. They're generally the same size and shape. And we just keep going down the line. Here's chromosome 3, chromosome 4, chromosome 5, etc. Right. So again, this is a karyotype, a photo of all the chromosomes sorted by size and shape and banding pattern. This karyotype is from a human cell. These human cells, we know, have 23 kinds of chromosomes. So in humans, that N number equals 23. We know that human cells, our somatic cells, are diploid. We're looking at a photograph of the chromosomes from one of our somatic cells. So in a human cell, we know they're diploid. We know N equals 23. So diploid again is 2N. We know N is 23, so 2 times 23 will tell us that there will be a total of 46 chromosomes in this cell. And if you count all the chromosomes in this karyotype, you will count 46. Again, 23 pairs of 2. So there's 2 of chromosome 1, 2 of chromosome 2, etc. So 46 chromosomes total in human cells. Again, we learned that by looking at karyotypes. That's how we could determine the N number and that our cells were diploid. And now we can use that information when we talk about human biology. All right, so just as a final reminder, these haploid cells are produced by meiosis, whereas diploid cells are produced by mitosis or fertilization. And so I just wanted to talk about the ways to make haploid versus diploid cells. So in this image, I'm showing you a bit of a life cycle. So we're starting here with some diploid somatic cells. Right? Inside each cell, there is just one kind of chromosome, and there's 2 copies of it. So both of these cells are diploid, and the N number is 1. These diploid somatic cells, some of them could go through meiosis and will produce these cells, which are haploid gametes. In the top, we've got an egg. The bottom, we've got a sperm. The egg has just one of the chromosomes. Again, there's one kind. In the egg, it's haploid. We only have one copy. The same is true in the sperm. There is only one kind of chromosome, and we have one copy. So we have made haploid gametes. We use these gametes during sexual reproduction to do fertilization. The sperm and the egg will fuse together to create a single cell that now has those 2 copies of the chromosome. So we just made a diploid cell, and we call that very first cell that occurs right after fertilization, we call it a zygote. So fertilization of 2 haploid cells together makes one diploid cell. And then we know that a zygote will develop into a baby, and then into a child, and then into an adult through many, many rounds of mitosis. Mitosis will duplicate these cells. We'll get lots of different cells, all of them identical to one another, produced by mitosis. So again, meiosis is the way we make haploid cells, and then we can produce diploid cells either through fertilization, combining 2 haploid cells together, or through mitosis, copying a pre-existing diploid cell. The last point I wanted to make to wrap up a conversation of haploid versus diploid cells is when precisely during meiosis are these haploid cells produced? So it turns out that cells go from being diploid to haploid during meiosis 1. So I want to point out this detail and then we'll end. So just as a reminder, here's a diagram of the full process of meiosis, where we're starting with a cell. We're going to go through prophase 1, metaphase 1, anaphase 1, and telophase 1. We'll do cytokinesis to generate 2 cells, and then those 2 cells will go through prophase 2, metaphase 2, anaphase 2, and telophase 2. And so I'm going to show you why meiosis 1 generates these haploid cells. So let's zoom in on just meiosis 1. So here's meiosis 1. Again, prophase, metaphase, anaphase, telophase, and then cytokinesis to produce the two daughter cells. And I want to remind you that during meiosis 1, the homologous chromosomes, and homologous just means the two of the same kind. So two of the same kind of chromosomes pair and separate into different cells. So let's look at that. Here it's easy to see during metaphase. This cell contains 3 kinds of chromosomes. There's the really large chromosome on the far right, there's a medium chromosome on the left, and there's this small chromosome in the middle. So 3 kinds, large, medium, and small. Again, there are 2 of each. We began as diploid. So there's one blue large chromosome, there's one red large chromosome. There's one blue medium and one red medium. One blue small and one red small. Okay? So that's what we began with in our cell. Again, the homologous chromosomes, the 2 of the same kind, are paired up during meiosis 1. And then during anaphase, we pull that pair apart. In telophase, we put each member of that pair into a separate nucleus, and then we separate the cells apart. And so let's look at the cells right here. This top cell contains 1 large chromosome, 1 medium chromosome, and 1 small chromosome. The other cell contains 1 large chromosome, 1 medium chromosome, and 1 small chromosome. So in this process, we started with a diploid cell, and we ended with these 2 haploid cells, 1 of each kind of chromosome. So it's during meiosis 1 that we create the haploid cells. And the one confusing point that some students have, I'll point out, is that they look in this cell after meiosis 1 and they go, wait, that large chromosome is made of those 2 sister chromatids. And I want to point out that we count chromosomes as 1 if the 2 sister chromatids are still attached to one another. So I'm going to add one point here, that when sister chromatids are attached to one another, they count as 1 chromosome. So again, that's why if you look in this cell that is made at the end of meiosis 1, we said that there's 1 large chromosome. That 1 large chromosome is composed of 2 sister chromatids, but we only count it as 1 chromosome. So I hope that helps with your understanding of what these terms haploid and diploid mean, how we can tell if a cell is haploid or diploid, and how we make specifically those haploid cells. So as always, let me know if you have any questions, and I'll talk to you again soon.